Hello, wonderful cell and gene therapy people. Welcome back to our shiny new webinar platform. This is where we'll be hosting all of our digital output going forward, including our sponsored webinars, working groups, and also breakthrough discussions like this one. The platform's been chosen because of its user-friendliness for the audience, its interactivity, ease of sharing, of supporting materials. On your right, you'll see a variety of options for reaching out to us via the chat and the Q&As. You'll also be able to see session handouts, which have some really interesting materials that are very relevant to this discussion and will be talked about during this discussion. You'll also see polling elements throughout this webinar. We're going to be asking you some pertinent questions to AI and cell and gene therapy. We're very excited to have a fantastic panel of industry experts here. On the 2nd to the 4th of September, Facilitate will also be hosting our Advanced Therapies Europe meeting for the first time in Barcelona, which is a blooming hub for cell and gene therapies at the moment. AI and machine learning will definitely be a topic of interest, with the panel covering its usage across the entire CGT work chain. We have a discount that is available until tomorrow. It's web off. It will give you a very generous 25% off registration. But today is slightly more focused on AI manufacturing, in a usage of AI in manufacturing. The overall focus we wanted this to be was the where, and the why of AI and machine learning in manufacturing sciences and technologies for cell and gene therapies. Really wanted to cut through the hype and explore what the actual use cases are for AI. So far, we haven't seen too many of these, and we're really excited to actually dig into this a little bit more, really cut through the bunk and see what progress has been made so far and where the promise is. Tiny bit of housekeeping, please include questions to Q&A. If we have time, we'll absolutely be asking these questions to our experts. And we're very excited to hear your responses to our polls as well. It is my pleasure to introduce Elaine Hamm, who is the Executive Director of the Alliance for AI in Healthcare, who will be moderating our session and also introducing our expert panelists. So, Elaine, over to you.
Great. Thank you, Ryan. I appreciate it. Hello and welcome, everyone. As Ryan mentioned, I am the Executive Director of the Alliance for Artificial Intelligence in Healthcare, or the AAIH, a global nonprofit advocacy organization that is dedicated to discovery, development, and delivery of better solutions to help improve patients' lives. And as you know, this session is titled Looking Beyond the Hype, because that's what we aim to do. We want to dig into what's real, what's working, what's maybe not working, and then, of course, what comes next as it relates to applying AI into cell and gene therapy. But first, I think we should level set, because AI is one of those terms that gets bandied about quite a bit. And so I'm going to give a couple of definitions, especially as it applies to cell and gene therapies. So AI is a broad umbrella term that includes everything from very simple rule-based systems to complex neural networks that basically mimic, human decision-making. Machine learning is then a subset of AI, where it learns patterns from data to make predictions or decisions. And then as it relates to gene therapy, it's often used for process optimization, quality control, or predictive modeling. But generative AI is probably what most people are familiar with. As we think about ChatGPT, it's a little bit newer, flashier, that creates new content, everything from text to new molecular structures. But what we're really focused on today is going to be applied AI, tools that reduce the development time, improve process decisions, and actually get deployed in these types of environment. Joseph was very kind in giving us a little bit more of a precise framework as it relates to definitions. There was a great article in Cytotherapy that was published by ISCT, and they really defined 3 types of applied AI as descriptive AI, so summarizing, visualizing data, predictive AI and forecasting outcomes, and then prescriptive AI, which is for recommending and automating decisions. So now that we got all of that out of the way, I will hopefully stop talking as much as we can turn to our incredible group of panelists. How we will structure the session, we'll try to have 30 minutes of panel discussion, 15 minutes of Q&A from you all. So we'll be able to put your comments and questions into the chat and read those out. And then wrap up with a lightning round of hopefully bold predictions of what's coming next. So with that framing in mind, let's meet our panelists. I'd love for each of you to briefly introduce yourself, who you are, what your organization does, and how you are utilizing AI or data in this space. And I'm just going to go down my list. So David, I'd like to start with you. There you are. Hi, David. Can you hear us?
Yes, I can hear you now. Sorry.
Great.
With the relation connection. So shall I introduce myself? Sorry, I missed
Yes.
Yes, so my name is David Sourdive. I am one of the two founders of Selectus. I am also serving as Executive Vice President for CMC and Manufacturing. We're a company that was founded a little over 25 years ago, specialized in gene editing. We were gene editing, and we were the first ones in that field. And today we deploy our know-how and capability in adoptive T-cell immunotherapy, essentially in allogeneic CAR T-cells with products today, essentially in liquid tumors, ALL, and ALL.
Great. Next on the list then is Irene. Please introduce yourself, what your role is, and how you're currently using AI.
Hi, I'm Irene Rombel, the CEO and co-founder of BioCurie. So I feel like I've spent the last 30 years getting towards this journey, starting in gene regulation, gene expression, and along the way, learning a lot of new technologies that are innovative, and actually spent the last 25 years at the nexus of big data and innovative technologies and advanced therapies. And so going from academia to investing, big pharma, biotech, now founded my own company back in 2021, along with my great co-founder, Richard Bratz from MIT, who's got a very complementary background. So I'm a biochemist and molecular biologist by training. Richard is a computer scientist and engineer. And so those are the kind of skills that we need to really make AI applied in cell and gene therapy. So I think that's kind of a good paradigm for all of us, that you need that deep subject matter expertise along with the nuts and bolts of doing AI. So at BioCurie, we're literally trying to transform cell and gene therapy process development and manufacturing.
Wonderful. Great. Thank you. Joseph, on to you.
Hi, Elaine. Thanks. Yeah, I'm Joe Egan. I'm a research associate, postdoctoral researcher at Teesside University and University College London in the UK. I've spent the past 20-odd years developing mathematical models in different areas, from epidemic modeling. I then moved to do my PhD in immunology, T-cell activation. And more recently, as my postdoctoral work has been in developing models for biochemical engineering, and particularly cell and gene therapy and CAR T-cell expansion. So I've been sort of developing these kind of real-time models. You might put them under the umbrella of predictive analytics. Whether it technically falls within an AI definition, possibly not, but I quite like that original definition you gave in terms of the predictive and the prescriptive analytics. Yeah, that's definitely where my work falls.
Wonderful. And of course, Kat, bring it on home for us.
Hi, everyone. My name is Kat. I am a scientist by training. In fact, my very first research project was in T-cell transplants long ago. I have spent the past 10 years commercializing various tools and technologies across modalities at Sartorius, Thermo Fisher, a number of small biotechs. I'm a consultant now. I am a strong proponent for a collaborative approach. I think there's a great space for us to do that in cell therapies in order to drive AI and ML-based analyses. And as part of that, it's implementing ethical treatment of the data and ethical AI.
Wonderful. Well, great. So we're going to first kind of talk a little bit about really sort of what is being implemented now, the hype versus the reality, as it were. So I'm going to ask this question to everyone on the panel. How are you seeing AI actually being implemented in the cell and gene therapy space right now, either from a technical side or maybe from a more administrative process operation side? Anyone, I can start calling on people. I can definitely do that.
I can certainly jump in from my own perspective. As I touched on before, we've been developing these kind of predictive analytics in terms of cells expanding in bioreactors. The particular project I've been working on has been working in a bioreactor where it's quite hard to take manual samples. And so really there's sort of limited information in sampling in terms of how many cells you have at a given time. So the question is, can we use the other data that's being received during the as cells expand, using PAT, so sensors for online sensors for say glucose and lactate, the perfusion rate. Can we use those kind of that high frequency kind of data to give you predictions of how the cells are growing? And so that's where the sort of predictive element comes into it. And then if you can predict that, then you can start to try to inform maybe the human operator overseeing the processes when might be the kind of optimal harvest time if you've reached your certain dose. So it starts to move from the predictive kind of analytics more kind of to a prescriptive. It falls under the name of a digital shadow. That's really a kind of virtual representation of a process. And it takes data in real time and gives you insight into the process and predictions in real time. That's slightly different to a digital twin, which some of you may have heard that term. And that's that whole idea that you're both taking data, you're making predictions, but you're feeding back into the system. That's something we're currently not doing. It's more just a shadow of the process, but that can still give you some insight. So that's what's one use case we've been developing for 3 or 4 years. There's a recent publication in the handout if people want to look into it in more detail. But that's something that's been quite useful for our industrial collaborators.
That's great. Kat, how are you seeing it implemented?
Yeah, I think there's some really interesting examples. I am to bring up to Lemmy Bio with the media development work that they're doing because they're handling some noisy data, and of course, noise is something that we're all struggling with. They're also tapping into high dimensional data, so I think there's some interesting stuff going on there. But I actually would say that we're under-hyping some of the AI capabilities. I think we're focusing on kind of some of the immediate applications, but there's just such a grander vision that we could adopt from kind of the AI forward space. I think we often talk about how we don't have enough data to execute on that. So I'd just like to bring up an abstract example. I had the great chance of talking to Frances West, who spent 37 years at IBM. She's a thought leader in AI. And she's talking about how she's Chinese. She wants to give that Chinese culture to her grandchildren. So she wants to create an AI agent who's Frances. And of course, she can't train from scratch Frances, right? There's not enough data that she can generate. But she can take an agent already trained on the basics of grammar, sentence structure, punctuation, etc., and give it the flavor of Frances so that her grandchildren can adopt the phraseology and so on. So I think that's the kind of thinking where we can take a model trained on vast data sets elsewhere and bring that into our space, which is very lean on data today. And I'm sure there are other folks doing work on this, but an example that came into my life was Curigen. So they're leveraging a model trained on basic fundamental biology elsewhere, but they're adapting it to cell therapy space. So I think that's just an interesting way of doing it.
No, love that. And we're seeing more and more of the models training new models as well. So, all right, David, on to you. David?
Yes.
Yes.
So for the lag. What we see in general is that the current application of machine learning essentially pertains to interpolation. We have to teach it on a learning set where we think all the precedent we will want to extract knowledge from are present. So we don't use today AI for extrapolation, or things unknown to the learning set. So one of the instances we see implement is the way we design DNA cutters, the molecules that we design for gene editing, that will recognize a very specific sequence, an entire genome. we use Talon. We've been using just about every kind of structure that exists. Uses home and nuclease, megatal, barrage, CRISPR, of course. But we focused on Talon because it's a protein action, and we can tweak it once we have identified a good locus, DNA, or modify. And that's a typical case where AI can be very helpful. Theoretically, there's only 4 modules that are needed to build a Talon: one for recognizing A, one for C, one for G, and one for T, the letters of DNA. In reality, we use 200 of them. We recognize DNA, unmethylated DNA. We use them not only as nuclease, but also as a base editor. We can go. All this is really enabled by the AI we implement, machine learning we implement. I shouldn't say AI, I should say really machine learning at this stage. And that allows us to optimize these molecular scissors that we develop. So that today we can strike 6 base pairs, basically, of any chosen nucleotide in any gene of any genome in any species, and eliminate off-target. That's really the beauty of it. So I think there we have a nice for AI with a set, some grammar, known enough of a learning set to actually produce that. We're also looking into other applications, and I know we'll be testing them today, where we also want to make our gigabytes of biodata speak more than they do and extract meaning from them. And that's something deploying and looking at seriously right now in process development, analytical development, clinical stratification, and also the stratification of the donors, the healthy donors that we to manufacture our allogeneic off-shelf T-cell products.
Great. And Irene, how about you?
Yeah, so I've been sitting here smiling because yesterday Kat said that this could get a bit controversial. So yeah,
we love that.
100%. So first of all, at BioCurie, we're actually doing some of the things that Joseph touched on. So we're actually developing digital twins, for example, for AAV process development and manufacturing, and applying some of the techniques that were mentioned and others. But first, going back to what Kat mentioned, underhyped. I think it depends on the context, because I tend to be a lot more cynical and think, gosh, there are a lot of pitfalls here, and there's a lot of, gosh, I think Ryan put it nicely, cutting through the bank. I mean, I'm usually not quite so polite, but there's a lot of nonsense out there. So I part of what I try to do is to set people straight on what are the pitfalls and limitations of AI, and then you can really understand the opportunities, go forward with your eyes wide open. So in terms of the hype, I think we're pretty much in the midst of a hype cycle, and I hope that it doesn't bite us, because that increases expectations, and people have it's almost like magical thinking, that AI is going to do all this amazing stuff. Well, no, there are certain limitations. So first of all, GIGO, this is something that everybody probably on the call is familiar with, garbage in, garbage out. So think about the data going in, propagating errors, you've got model collapse, you've got underfitting, overfitting, data bias, you've got outliers, hidden variables, and also remember, some of the data that you're feeding in could be based on poorly designed experiments, right? Generative AI isn't going to tell you if that experiment was properly carried out or not. And all of us have read papers that are just rubbish and should never have been published, right? So you need some degree of curation, and human curation is expensive and it requires subject matter expertise, ergo most of the people on the call. The other thing is, and this is something that David touched on, machine learning and traditional AI can't extrapolate. And that's where, at least at BioCurie, we're pulling in that mechanistic piece of the modeling that's relying on fundamental biology, chemistry, and physics. And then we're not just hybridizing, we've actually got proprietary ways of integrating that with AI. And that way we can extrapolate, because everybody has to keep in mind that on the call that data can't predict or go where it hasn't been, right? So if the data is here, say you have patients with a certain phenotype, that you want to predict, if they're not in your data set, you can't possibly predict how they're going to behave in a clinical trial, for example. So, and I've actually heard that suggestion from non-subject matter experts that are very smart in AI ML, and I'm like, oh my gosh, this is why subject matter experts are so critical to make sure that we keep patients safe.
Yeah.
Another caution is black box models, and I'm sure many of you all know that FDA is not a big fan of black box models. So now we're moving more towards interpretable and explainable models where you can actually draw, a line between the math and the actual, underlying biochemical principle, for example. So that's one, it's a lot safer, but two, it means that you can make a lot more intelligent predictions. So those are some of the main points I think that people should be cognizant of, but especially it's not a magical salve for everything. It's a very, very useful tool, and eventually we will be able to do more and more powerful things, but we have to walk before we can run.
Absolutely. And those black boxes are quite complicated, and I don't know if we're going to get to a glass box anytime soon, but maybe we can start working more towards an opaque or gray box. And when we start sharing more and getting out of our silos, we can make it a little more transparent. But Irene,
yeah, translucent's a good start.
Yeah, we'll start there. Well, Irene, I'm going to stay with you. Can you share a practical example of a way that you're employing it? And you talked a little bit about the hype piece of it. Let's talk a little bit about how you're actually using it.
Absolutely. So at BioCre, what we're trying to do is really democratize AI-driven decision making, and with the realization that there are very few people with that subject matter expertise in AI and ML, as well as, say, cell and gene therapy, for example. And there frankly aren't enough people in the world to do all that. We're building a platform where all the decision making's been and intelligence has been built into the software. And so there you can have an end user such as an engineer or a biologist, that can do millions and millions of basically in silico experiments to optimize the process, for whether it's for AAV or whatever that modality is, and be able to apply that during process development and subsequently to manufacturing. So that's essentially what we're trying to do, to democratize that. And to do all that requires a heck of a lot of complex implementation of proprietary algorithms to do all those, all the different steps that a whole team of AI experts would do.
Great. Joseph, I'm going to ask the same question to you.
Yeah, thanks. I've kind of touched on the use case that we've been developing for the past few years now already. But I think it's interesting, that whole idea of, my background, as I said, is more on the mechanistic side of modeling, which has been around, frankly, for hundreds of years. It's only in the past 5 to 10 years that this new term AI has come, and this new form of predictive modeling has come on board. Because of the explosion in data and the explosion in computing power, which has allowed these new forms of they're more statistical models, if you think about machine learning and the models, finding patterns in those data, but you need a lot of data. The advantage sometimes of having these mechanistic models is, one, they're explainable. So under the framework of AI, they're kind of explainable AI. Often they're simpler. You have fewer parameters in there, especially if you can simplify down the mechanisms of biology. Sometimes you only have a few parameters to fit, and then sometimes you don't need as much data. So there's benefits to both. And I think Irene touched upon what's quite nice in terms of how the sort of mechanistic modeling has evolved alongside this AI, is that you're starting to see these hybrid models. So you can start to kind of fuse these mechanistic ideas that you really understand quite well. And then there are the areas where you don't understand, and you're sort of being guided by the data. And often it seems that when you combine the two to form these kind of hybrid models, then you start to get the best of both worlds. So I think that's where we're trying to head. I always have a background and a passion for the mechanistic modeling. That's my bread and butter. But I think I've started to notice that actually we can enhance these approaches using these new AI ideas. And sometimes you have to just use AI because you don't understand the mechanistic principles. But if you do, then, combining these two is, I think, a really great way forward.
Yeah. So again, if you don't mind, a little bit of controversy. So first of all, AI has been around since the mid-1950s, except most of us haven't heard about it until maybe the last decade or so. But for context, it's actually gone through several summers and winters. And so it's gone through a number of hype cycles. And I just hope that, because of all the current hype, that it's not going to set us up for, great despair afterwards because of misapplication. So hopefully this one will be the real thing that'll be sustainable. So that is one thing. The second thing, 100% agree on mechanistic models and how fundamentally useful they are. Unfortunately, they're not so simple always, and in fact can be extremely complex. And there's one that comes to mind, just for example, the model for mechanistic model for AAV production in SF9. So that was developed again at Richard Bratz's lab by Francesco Desjo. And in the very first step, he had to do 200 ordinary differential equations. And that's just to get started, right? So there's nothing simple about some of the more, the mechanistic models for more complex modalities. It's a little bit different from something, classic example is force equals mass times acceleration.
Yeah.
Oh, sure. You can make mechanistic models as complicated as you want to, but yeah, I always try and drill down to the fundamental things if possible. But as you say, sometimes it's not possible.
Well, and David, I want to stick on something that you mentioned when talking about data and shifting over to data, because that's a lot of the foundation of what we're talking about. And I'm going to open this question actually to all of you. So feel free to raise your hand on this one. So I'd like to understand what kinds of data that you think is most valuable for AI or ML and as it relates to cell and gene therapy, specifically, whether it's process or omics or imaging, et cetera. And then what types of pitfalls are you seeing companies kind of fall into in that respect? Who wants to go first, or I'll start? All right, David, please, you raised your hand.
Okay. In our experience, the limiting element today for cell therapy is very much in the analytical side. The metrics that we have that are available to describe cells and cell populations are limited, and they're not always relevant. We use a lot of cell surface markers, CD something, CD8, CD what have you, that speak actually more to the cell's ontogeny than it does to its role in a particular scenario. Like, for example, if we compare the blood of many healthy individuals, all the people around this call, and we take a picture with those metrics, and we take another picture 6 months from today, it will all be very different. We'll all have distribution all over the place. However, functionally, will basically work the same. If we catch the little virus of the moment, it's going to react the same way in the same scenario. So functional similarity is not correlated with the structural similarity as we measure it today. So the problem we have lies in the metrics. So we can accumulate a lot of data there, but it doesn't speak to what we want to see. So that's a problem. And that actually expands beyond just the metrics of the cells. It's a lot of biodata is like that. We have the probes and we have the means to measure what we can measure. That extends to genome sequences. Today, we cannot predict, reading the whole genome sequence of a species or a variety, how it will or will not behave. And if we want to go further into genome design, it's a black box, because the genome was never written by humans to be read by humans. It's not cryptography. It's an accumulation of attempts over billions of years. So it may never be readable by human. It may be readable, quote unquote, readable by AI. So I think I think that's one very important element. What is it that we're looking at, and be humble enough to realize and recognize that the metrics we have today are limiting, and they do not incorporate the knowledge, the grammar, what is filling the gap between information and meaning. The second problem, I think, is the size of the learning sets, because of the little that we know and all the unknowns and the variables. As a consequence, we need huge learning sets, and biodata in that sense is quite different from what we had in other fields of application, and that's very impractical without some grammar. So we need to move forward combining knowledge with a more agnostic approach. So it's a sort of back and forth, and the AI we use in our implementation is a bit different. So fulfilling that big promise that with AI we're going to be able to make all the biodata speak is I think a bit, we have to be cautious. So I'm optimistic with caution. and I think there are instances where we're getting there, like standardization of cytometry is one of them. There's other ones that are quite
100%.
But it's going to be gradual.
Right.
We
Right.
need
So
to do the work, and I
Oh, I think we might have lost David.
yeah.
Sorry, David, we're having slight technical difficulties with your mic. Sorry, I think there's just a slight connection issue there.
Yeah, so Elaine, in the meantime, if I could pick up from where David left off. So part of it is initially, you don't know what those key variables are that are going to be the most important elements of your model. But then again, you can't boil the ocean and try and can you see me? I've got an error message. Can you hear and see me?
Yeah, we can see you.
Okay, great. Okay, fantastic. Yeah, so until you know that, you can't just collect everything and at great frequency, because that's just prohibitively cost expensive, and it's not economical. It's not economically viable. So you kind of have to initially, we're starting off with what I discovered, to my disappointment a few years ago, is we're currently, and then for the foreseeable future, going to be stuck with small, sparse, crappy data sets, right? We're not going to have what is called big data in any sense of the word.
No.
Foreseeable future. So we better get pretty damn good at being able to do very highly predictive modeling with what we have. But for starters, having time series data, that's helpful for certain things, but only for the parameters that are really useful. So again, it all depends on the context of use, right, and the application.
If I could jump in just in terms of what I've been working on in terms of, say, bioreactors, sometimes if you're able to implement the PAT, the process analytical technology, and you're able to automate it as well, then suddenly that opens up the ability to have some quite high frequency data. Now, yeah, sure, to begin with, we've been monitoring, say, glucose and lactate online and getting high frequency data. Perhaps as you start to do metabolomics on sort of samples throughout that process, you can start to identify some other key pieces of information that in the future you might want to develop a sensor for, so you can monitor that, and that can take time. But if you start to get that data, then that opens up the possibility to get so much more insight. And yeah, I think as more kind of, my field, more bioreactors have kind of come online, over time we're starting to see more data come in. I mean, dissolved oxygen is one of those standard ones that is often there, but hopefully as more and more bioreactors come on board, then we'll start to get both automation and that process analytical technology. And that just, when we get that data, suddenly it opens up the ability to do modeling, to do AI, because without that foundation, you've got to remember that the ability to get that data, in my field, it's the PAT, it's the automation. Without that, you can't do any AI. So you have to get that in place first. And hopefully build up those sensors over time when you start to realize, you start to identify those critical process parameters and critical quality attributes, and you know kind of what you need to monitor, then you really start to make some progress.
That's great.
If I may add to that, I think cell therapies are unique in that sense, where let's say for other modalities, we have so much better defined CQAs that it's easier to do a contained, well-characterized run that ends at your product characterization. Well, for cell therapies, I would argue that we might need, at least in the beginning, to go a step further and look at patient data, look at those clinical outcomes, which then really constrains those analyses to only clinical trials. And maybe one day when we can correlate preclinical models to clinical outcomes, maybe to also preclinical runs. That means technology companies just can't do this on their own. These data sets, technology companies perhaps never going to run a clinical trial. This is with a therapy developer. Also the ethics question to that, right? Anyway, so I think in that sense, we're in a very interesting spot. And I would argue that in the current economic climate, we're not in a position for any one set therapy developer to do that massive experiment with great characterization, wonderful omics. I know we all dream about this, but I think practically speaking, it's just too expensive right now for any one company to do that. So that's where I think there's that opportunity to collaborate and in a confidential way with actually sharing the data, but look at what companies have seen in the data that they already have, because these are just such expensive data sets to generate. You're on mute, Elaine.
Sorry, I had a pop-up button come on, so I was trying to explore the chat to see if we had any additional questions, and then I turned off the mic. So, well, great. I also, sticking with the data theme, I'd like to better understand where you think there's gaps, and then maybe sort of on the opposite side of that question, where is there too much data? In the sense of where you have a lot of information, but what do you do with it per se? I mean, I see that a lot. I mean, like, great, I've tracked my steps. What am I going to do with that information? I'd like to understand sort of where you think the gaps are, and maybe a little bit where the glut is as well.
Okay, Elaine. So I'm a huge cynic when it comes to just stamp collecting data and collecting data for its own sake.
Yes.
I don't think I've seen any glut of data in the cell and gene therapy space. If anybody has seen it, please share, because I don't know where that is. But it's certainly in other spaces. I agree, you don't just collect data for its own sake, because it's got to be actionable. Of course, a priori, we don't necessarily know what that actionable data are, as Kat mentioned. We don't even know what our CQAs are for some of these new modalities yet. But then again, all of these, every data point has some cost associated with it. And so ultimately we have to be able to justify an ROI. And that's something that nobody's mentioned on the call today yet. This is not free stuff, right? AI is quite expensive in terms of compute time and even electricity. So, we've got to be really mindful. Yep. There are a lot of costs, cybersecurity, et cetera, and it's a whole other conversation. So, this is just because even if it's in the cloud, it's not free.
Any other comments on that?
Yeah, I think ideally, if you're collecting data, ideally you might want to perform a design of experiments. So you kind of
have a hypothesis.
Cover quite a big area, and again, that it can be difficult. It can be expensive. The paper we were working on, we had a certain number of data sets. They were difficult to generate. They were expensive, and in the end, we had to do what we could do with the data available. I mean, I think fortunately the approach we took helped that, because it was a simple model with mechanistic, only a few parameters, we could get away with it. So yes, ideally we're looking for, covering that data space by design of experiments and other techniques. It obviously is not always possible, and so sometimes you might just have to rethink your approach, possibly with the data you have. Great.
Well, I'm going to switch over to the Q&A put into our chat box by our attendees. So I'm going to start with Chesney, who was first. How is application AI ML being applied to process development, which is very variable as we develop and optimize manufacturing processes? Anyone want to take a crack at that?
Sorry, I had a pop-up happen.
Ryan, his mini surveys here.
Yeah, we're all getting the same surveys. It's little.
Speaking of data gathering, of course. So
I'm trying to make sure they pop up to disrupt you as much as possible.
Yes, thank you, Ryan.
That reminds me what the question actually was.
What's happened? Caffeine. Okay, how is application AI ML being applied to process development, which is very variable as we develop and optimize manufacturing processes?
Yeah, so that's exactly what we're doing at BioCurie, to take out that undesirable variability in the process, because that again is one of the unfortunate nuances of advanced therapies, and that it's harder to get that good high reproducibility every time. So that is something that AI-driven modeling can certainly reduce. On the other hand, there is the desirable variability in that you can be very flexible with the variables, because of course, in contrast to going back to the lab, or especially doing DOE with different variables, you can virtually test really an unlimited number of variables in silico if you've got the right kinds of predictive models, and you've already got enough. And so again, we've got enough data previously to be able to deal with certain variables. So then we could very, easily do those in silico, as opposed to, the good old fashioned, rolling the sleeves going into the lab, which is why I love AI so much as a biochemist, because I used to do it the hard way.
Yeah, right, exactly. Anyone else want to comment on that question, Joseph?
Yeah, not to add much. I think it's a good question. It's a really hard one to answer when you don't have that much data. What can you apply some of these tools? It is a real challenge. I mean, I've been lucky enough to have some data from 17 different runs from a DOE, and by combining those data sets, or fitting the model to combine data sets, I've been able to tease out a little bit more information than I would have by just fitting to the individual data sets alone. But I do appreciate when you're doing those process development and those experiments, it can be a real challenge to implement some of these tools.
Wait, I'm going to switch over to another question by Thorsten Gorba. Subject regulatory limitation. How can we convince HTA to take out the human in the loop? Oh, where'd it go? That question just disappeared.
I think it was something about fully autonomous, maybe decision making, something.
So
I can.
There, it's back again. Sorry, got it. All right, human in the loop for AI, ML driven process decision making to arrive at true autonomous live data or inform autonomous operation of automated manufacturing processes.
I can just briefly step in there. I mean, I think it's a good question. I think if you look at some of the regulatory documents, we have them in the handouts, some that have come from the EMA, the FDA, they talk about risk-based assessments. And I think, sometimes you're looking at sort of low-risk AI, and sometimes you're looking at more high-risk AI. And I think as soon as you start to take the human out of the loop and you have, say, fully autonomous kind of AI kind of making decisions, then you're obviously moving much more towards the high-risk AI, and that's going to require greater regulatory oversight and everything that comes with that. I think, some of the things we've been working on, in some ways, we ideally want to keep the human in the loop. We want to keep it low-risk AI. We want it to be an informative tool for that operator overseeing the process to improve their decision-making. So I think that's why it's a digital shadow. It's not a digital twin. It's not taking control. Maybe in the future you can get to that. But at the moment, we see it as a bit like AI is a tool for us as we're doing our work using ChatGPT. It's a useful tool. Sometimes we can think about using these AIs as useful tools to improve decision making and keep the human in the loop. So that would be my take on things.
Yeah. So actually, along those lines, the more conferences that I've been going to recently on the data side, the more I'm hearing the inclusion of human in the loop. So I think we're moving back towards keeping a human in the loop. And keep in mind, Biopharma 4.0 is the no human, 5.0 is with human. And I think it's become increasingly important to keep that human element. But that said, even with the recent FDA regulatory draft guidance, I didn't see any explicit request to keep the human out of the loop. I don't know if anybody else noticed it, but I did not see that. There are a lot more cautious about the nature of the data, how you validate, how you trained, validated your model, lifecycle management of the model. So having whether or not you have a human in the loop, that's not just an AI question. That could be any kind of model-driven automated systems that are used during GMP manufacturing, and there are a whole bunch of FDA guidances around that already. So we're not creating anything new. I mean, the fact that you're using AI instead of some other modeling approach, I don't think is the salient difference necessarily.
Yeah, if I may weigh in on that one. So I think sorry, just another pop-up. I think that this challenge that we have with kind of how do we control that the outcomes, the insights, suggestions from AI are high quality. This is not constrained to manufacturing or pharma or healthcare. This is across all regulated industries where the impact of, I would call this negligence, right? But some bad suggestions that the ChatGPT gives you for dinner tonight, not an issue. You just have maybe one bad dinner. Hopefully you don't end up in the hospital with a seafood allergy, right? But other than that, it's okay. If you have a high impact decisions that you need to make, and I'm going to abstract away from healthcare, just the big picture, right? For who gets to get the mortgage loan, what cases you put into your lawsuit that you quote, right? All of these things, this is true for all of us. And I would love for us to see a more combined effort across all regulated industries, because it's a shared problem. Once again, free competitive, right? But so the current trends that we see is this human in the loop solution, where, this is the proposal that has been made for regulation of AI in the Senate by a Massachusetts senator. To me, that's a temporary fix. Yes, today, so we because we can't control the quality of the insights, we have to have a human sign off on it so that take legal responsibility. But I would hope that we can think of other ways of implementing that legal responsibility and getting the algorithm actually involved, somehow motivating the algorithm to produce better outcomes. I have a framework in mind. If somebody wants to talk about this, I'd love some input on this, some thoughts. And then to address the earlier point about black box, I think that there's a chance that we won't get past the black box level insights. I mean, we've been struggling with this in neuroscience. I went to school for neuroscience, so I can speak to that. We don't really understand how the box produces the thoughts, yet we do still have the thoughts, right? So can we have, at least in this temporary stage, can we have some level of black box readout for the goodness, the ethics, the reliability of the insights? A way that I think about it, again, would love to get some thoughts from folks on the call, is how we think of psychopaths, where they have a brain that produces very different insights from kind of how the rest of us operate. We don't understand how the brain works, but we can see that on an fMRI signature. Can we apply the same level of black box readout for AI algorithms to have this, let's say, continuous certification for the reliability of what they offer us?
I think it's a really important distinction in that you are, thinking about black box. Well, there's a lot of drugs that we don't exactly know how they work, but indeed they do work. And so you don't want to, I guess, throw the baby out with the bathwater just because you don't have a complete insight into it. But of course, you have to be careful in terms of transparency and explainability as we move into the regulatory risk mitigation piece of it. All right, I'm going to get into a couple other questions here from the panel, or from the audience before we start to wrap up, because we're already almost 10 minutes before this ends. So a lot of stuff to cover. All right. How long do you think it will be until personnel working within the biomanufacturing sector will need to be trained on and understand how to use the work with AI? Great question.
Well, I would say that today everybody should have some basic fundamental understanding of AI. It's like using the internet. I mean, nobody thinks about it now. We're using an iPhone, and yet remember, it's not that long ago when it was this new thing, and we were just trying to wrap our heads around it, right? So it's just going to become, so just to actually, this is the quote that Ryan and I laughed about, because it was my quote that was attributed to generative AI, but it's about, scientists with AI will replace those without it. It's true. It's just another tool. So whether you're a process scientist or in discovery or wherever you are on that across the, end-to-end spectrum, you should be using AI at, to some level, and at least understand some of those limitations that I mentioned, so that you can use it cautiously. It doesn't mean that you're necessarily going to be using models for AI-driven decision making, but you can't really use your computer during the day without having AI influence you.
If I could just jump in, my comment also kind of relates to the previous question. I think a lot of this will come down to the trust and acceptance. And we need to think about, transparency. We need to think about the transparency as much as we can in the sort of model architecture, the transparency in the model performance. And we also really need to think about transparent communication to those human in the loop, those human operators sort of overseeing the process, using these tools. We need to be really transparent in our communication, because without that sort of trust of a tool, it may well be ignored or just not used. And so, I think the people working in this industry, we really need to take them along this process, training, communicating, explaining the limitations and the pitfalls, and that it's not a silver bullet. It is just like all the other tools that you might use. It can help, but it's not going to solve everything.
Sorry, maybe just to weigh into that. To me, one of the basic principles is keeping your company's information confidential. Whatever AI tool you're using, please just don't upload any confidential data into that. If it's ChatGPT, I know the temptation is vast, but please keep in mind this has profound impact on IP know-how. So it's a safety of implementation of AI. I think it's just a key part of this training.
I think that's something that companies don't think about when they're starting to look at how to implement AI within their organization, is some of the more legal risks that come with it. And we started to implement having our consultants, consulting agreements, not, you can't upload our data into XYZ types of programs. So
exactly.
Yeah. Great. Let me move on to the next question from Lan. What types of AI implementations are common in cell gene therapy domain currently? Are they primarily traditional, generative, or agent-based systems? David, you want to?
Maybe I can start.
That'd be great.
Yes, maybe with this. So what I described earlier that helps us design the gene editing component of our manufacturing and design is, I would say, semi-classical, although there's a bit novelty in it. What I see now more and more developing, and this is true in many different platforms, is that agentic AI seems to be quite popular. And this means that there's going to be an element of customization. We're not going for the big global giga model. It's going to be a sort of do-it-yourself or mix-it-yourself approach. And every platform likes to have their own mix and blend of different AI agents. So it seems to me that we're not heading towards a standardization. There will be multiple kinds of outcome when prompted with the same data set, and we will have to live with that. So I think there's going to be plenty of work for validation, for burden of proof. What we need is, of course, a few successful precedents. That's really what the industry needs. But it seems to me that every time we're going to be using AI to back any claim we make, we will probably need to come with data generated with different configurations and not just one model, to show that n equals a lot still means something, and that there's a reasonable conclusion we come to. I think that's really what I'm seeing right now, and there's a lot of players around that help and support this industry by customizing an AI approach. They don't come with something on the shelf and say, oh, this is the model everybody's going to use. It's so as soon as you do that, you do generate different configuration, different model, de facto different models, and this means there's going to be a lot of distributed work to make those accepted and recognized.
That's great. Well, we're coming up to about 5 minutes left, so I want to make sure that we get into our hopefully bold predictions as to where we think AI is going to go in the future. Thank you to our attendees for all of the amazing questions that we have in there. I think we might, I don't know if the panel would be willing, but I think it would be great to be able to take these questions and some of the things we've talked about and turn it into maybe a potential follow-on publication. That's something that we do a lot in the AIH is making sure that we can capture the things that we talked about in this great panel while we have the experts gathered here and in the audience to be able to proliferate this knowledge. But before we close, I'd like to know everyone's bold prediction that you have for the future of AI, specifically in cell and gene therapy in the next 5, maybe even 10 years. David, we'll stick with you and we'll go around the bubble screen here.
You're on mute.
Oh, you're on mute, David. Sorry.
Sorry. I think we will see some precedents of successful deployment in specific use case. 10 years is both a long and a short time. If you think of regulatory timelines, it's going to start becoming a more regulated, more better understood approach by regulators, but it's still going to take a bit of time. What I anticipate, though, is that we will have fast and easy results on low-hanging fruits, and then something that's going to not necessarily be a plateau, but it's going to be a more gradual, incremental improvement of our activities thanks to AI, because we need to generate the data. That's what we said earlier. So low-hanging fruits in the first coming 5 to 8 years, and then we're going to be on a maybe less steep a slope. And what I anticipate is that we will have, in some cases, excellent precedents that will help everybody change their mindset and appreciate the benefits we can have when the problem is amenable to AI, when the data sets is there, and when it's a nice play for such an approach.
Irene, let's hear your bold prediction here.
Yeah, so this is bold, and this is literally what I'm, betting my life on is that we'll be able to leverage AI to really have a profound improvement in speed and safety and cost of cell and gene therapies for patients, really to have that big step change that we need to bring these therapies into mainstream medicine, because today the business model is unfortunately still a bit broken. So my prediction is that AI will be one of the key technologies that helps bridge that gap.
I love that. Joseph, all right, you're next.
Yeah, it's a brave person who makes predictions about where AI is going in the future. But,
always.
But I guess from my point of view, yeah, I'd love to see more automation, more process analytical technology, so we can gather that data, so we can start to implement more of these tools. I think, there's probably going to be quite a lot of sort of low-risk AI that makes a lot of improvement, like David said, in the next 5 years. And then it's, those kind of high-risk AIs is going to be, obviously, they're going to be harder to get through. But yeah, we'll see.
That's great. And Kat, last but of course not least.
Yes, I am a technocrat at heart and such an optimist. So what I hope and believe that we can do is get together, especially given the current economic climate, be more collaborative, be more cost efficient through collaborative approaches. So what I think can happen for us is that we have, every company has its internal AI agents who are experts in upstream, downstream, this modality, and so on. You can think of them as your expert Bob, who has been at the company 30 years, knows everything about your process. You go to that person or agent to troubleshoot. The collaborative piece, to me, is an opportunity for us to set up a framework for these agents to collaborate in the digital space confidentially and share industry best practices, much like our experts do when they go to conferences. But setting up that same exchange in the digital space in compliance with the legal wishes and requirements, right? Doing this in a way that we're not sharing data, we're not really sharing the insights themselves, but we're enabling that collaboration so we can get to answers faster and at a lower cost.
That's great.
Yeah, if there are any lawyers on this call, they're probably having a heart attack right now.
They are. That's fine.
I've been talking to lawyers about this, Irene. I think there is a way.
I mean, sometimes with boldness, there might be some laws broken, but that's all right.
Fully legal. Fully legal. It's going to be great.
It's going to be great. It's going to be fine. Well, there's one thing to take away today is that AI is not magic, but it's probably not optional as we move into the future of drug development and healthcare. But it's a tool, and like any tool, it depends on how well we are preparing the foundation, our data, our systems, and of course, our people and our organizations, as well as probably a little bit of our expectations as well. So I'd like to thank our amazing panelists for all of your insights and for all of you for joining. If you would like to keep the conversation going, hopefully we can maybe come up with a publication. I can wrangle this group to come up with a publication, and we can continue this this on a paper, and feel free to reach out to connect with any of our panelists as well as myself, and connect with the Alliance for Artificial Intelligence in Healthcare. That's perhaps maybe where we'll be able to put that paper. So thank you all for your time, and have a great rest of your week.
Thank you.
Excellent.
Bye.
Thank you all for joining. Thank you so much for joining. I did find out how to make those polls less disruptive right at the end for the very last one. So that's a good lesson learned for next time. I really want to thank particularly Elaine and our panelists. I'd be very keen to hear what you want to hear about next when it comes to AI and cell and gene therapies, and what part of the workflows you want to talk about when it comes to this. That poll was quite useful for that for us. We'll also be looking to launch a working group on data standardization in cell and gene therapy. Again, one of the polls was indicating your interest in participating, so very glad to see a great response to that as well. Or get in touch with me directly if you do want to be involved in any way. As I mentioned at the start, if you want to join us at Advanced Therapies Europe on the 2nd to the 4th of September in Barcelona, please use the discount code, which is available until tomorrow. And please look to the event agenda, where you can see some of the program that does talk to AI, manufacturing, automation, and analytics in cell and gene therapy. And yeah, thank you again, Elaine, for hosting this particular panel. And to all our lovely panelists who so generously shared their experience and opinions on this timely topic.
Thank you.
Thanks, Ryan.
Thanks.
Thank you.
Thank you.
Bye.